For part 1 of our experiment, we had to model the breaking of DNA molecules by restriction enzymes and the gel electrophoresis process of those DNA fragments. The first thing we had to do was to assemble the actual DNA molecules based on the different cards we got. One of them was for the crime scene, one was for the victim, and the other 4 were the different suspects. We had to use the different colored pop beads to assemble the DNA molecules. The red pop beads represented phosphate, the white pop beads represented the deoxyribose sugar. The orange pop beads represented thymine, which base paired with the yellow pop beads, which were adenine. The blue pop beads, which were cytosine, paired up with the green pop beads, which were guanine. And we had to connect these base pairs using the plastic connectors, which represented the hydrogen bonds between the 2 different nitrogenous bases on the different strands of the DNA molecule. So once we were done assembling the DNA molecules, they looked like these. Once the DNA molecules were assembled, we had to use the restriction enzyme cards to cut the DNA molecules into specific fragments. So we first used the JAN1 enzyme card to locate the specific sequences that the enzyme recognizes and break the DNA molecules at those sequences, as you can see here. Then we did the same thing with the Ward2 enzyme card, except the Ward2 enzyme card recognized a different sequence than the JAN1 enzyme card, so we broke the DNA into further fragments so that they looked like this. Once the DNA was broken down into specific fragments, we had to model gel electrophoresis by creating rows and columns on the laptop, where then we simulated electrophoresis as shown by moving the DNA molecules from the negative to the positive end based on the number of base pairs that the DNA molecules had, as you can see in this clip. What we then had to do was use the alkali cards and remove the bottom strand from the DNA sequence. So, as you can see here, the alkali cards recognized certain sequences of base pairs on the DNA fragments, and so we split the 2 strands when the alkali cards recognized the specific locations on the molecules. Then we used the probe cards, which also recognized certain base pair sequences on the DNA fragments. And all we simply did was line them up with those specific sequences that they recognized to represent the idea that those were the amplified areas on the DNA molecules that would be further studied in an actual gel electrophoresis experiment. Once we were done with all of the parts of our experiment, these were the results that we had. For part 2 of our experiment, we had to conduct the actual agarose gel electrophoresis process, in which we inserted the DNA samples into the agarose gel and watched as they migrated from the negatively charged side to the positively charged side. As you can see here, before we started our actual experiment, we had to boil the agarose down into a liquid form so that we could easily pour it into the casting trays. Once the agarose was boiled and the casting trays were made, we simply had to pour the agarose into the trays and then store them in the refrigerator so that they solidified for when we were ready to conduct the actual experiment. For our experiment, we decided to use the submarine method of loading the DNA into the agarose gel. So in this process, we had to place the casting tray into the chamber, and then we poured the buffer solution into the chamber just so that the buffer was just a little bit above the level of the agarose gel. Then we used a micropipette to put 10 microliters of each DNA sample into the wells created in the agarose gel. So in the first lane of the wells, we had the DNA marker standard sample. In lane 3, we had the crime scene DNA sample. In lane 5, we had suspect 1 DNA sample. And in lane 7, we had the suspect 2 DNA sample. As you can see here, this is where we started to actually watch the DNA samples migrate from the negative side to the positive side. So in order to do this, we had to connect the chamber to the power source using the different black and red cables. And then, once we turned on the power source, we just let the chamber sit and we waited until we started seeing the different fragments of the DNA samples actually start to move based on length. What we then had to do was turn off the power supply once the DNA fragments were done migrating to the positive side, and we had to put it in the staining tray, and then we poured the DNA staining solution over the gel so that it looked like this. What we then had to do was, in order to remove the stain so we could just see the DNA fragments in the gel, we had to continually pour in water and drain out the dilute stain that way. It was a very tedious process, but we got it eventually to look like this. Finally, as you can see here, we measured the distance from the wells to the bottom of the DNA fragments, each one from each sample, using a ruler and measuring that in millimeters. And then we plotted it into a table and later on a graph, which you will see in the data and results portion of this presentation.